Good morning, grade 9. Today we will start the last section of chapter 6. This is 6.6, which is called Meiosis and Genetic Variation, page 179. Our key concept today is independent assortment and crossing over during meiosis result in genetic diversity. We have two main ideas. Number one, sexual reproduction creates unique gene combinations. Number two, crossing over during meiosis creates genetic diversity. Let's start by the main idea number one. Sexual reproduction creates unique gene combinations. The major advantage of sexual reproduction is that it gives rise to great deal of genetic variation within a species. Like this picture, you can see how the people are very different from each other. This variation results largely from number one, the independent assortment of chromosomes during meiosis. Do you remember the meiosis? Yes. And number two, the random fertilization of gametes. Recall that homologous chromosome pair up randomly, the homologous chromosome pairs, pair up randomly along the cell equator during meiosis 1. In other words, it's a matter of a chance which of the two chromosomes from any homologous pair ends up on a given side of the cell's equator. As you have learned, human cells have 23 pairs of chromosomes. These are the 46 chromosomes of human cells, and each pair line up independently. As a result, gametes with 2 to the power 23 or about 8 million different combinations of chromosomes can be produced through meiosis from only one human cell. Now think about the fact that the sexual reproduction produces offspring through the random combination of gametes. In humans, for example, a sperm cell with one of 2 to the power 23, which is about 8 million chromosome combinations, fertilizes an egg which also have the same number of chances, the 2 to the power 23 or the 8 million chromosome combinations. Since any sperm can fertilize any egg, so the total number of possible combinations is the product of 2 to the power 23 times 2 to the power 23, or more than 70 trillion possible combinations. In other words, any human couple can produce a child with one of about 70 trillion different combinations of chromosomes. This is a huge number. Independent assortment and fertilization play key roles in creating and maintaining genetic diversity in all sexually reproducing organisms because of this number of chances. However, the number of possible chromosome combinations varies by species, so the number of possible combinations vary by the species. The probability that the bald eagle, this is the bald eagle, or the rabbit-eared bandicoot, this is the rabbit-eared bandicoot, will inherit a specific allele is determined by the same way that is for the pea plant. Sexual reproduction creates a unique combination of genes. This results in organisms with unique phenotypes that are the physical appearance. The offspring of sexual reproduction have a mixture of both parents' traits. As we said, traits are inherited from both the mother and father. For example, a rabbit-eared bandicoot offspring all share many traits for the things that make them bandicoots for the same species, but they may also have many different, or they may differ in many ways. Like what? Some may be colored more like the mother, others more like the father. Some may dig deeper burrows or hunt more skillfully. Others may in time produce more milk for their own offspring or have more litters. Having some of these traits may allow one bandicoot to reproduce in conditions where other bandicoot could not. The second main idea is crossing over during meiosis increases genetic diversity. It's clear that independent assortment creates a lot of variation within a species. Another process called crossing over occur during meiosis and help create even greater variation than the 70 trillion possible combinations, as we said. Crossing over is the exchange of chromosome segments. Segment is a part of the chromosome between homologous chromosomes during prophase 1 of meiosis 1. So the process of crossing over help in genetic diversity. It happens or take place during prophase 1 in meiosis 1. What happens exactly? It's an exchange of chromosome segments, part of the chromosomes. They are exchanged between the homologous chromosomes. At this stage, each chromosome has been duplicated. The sister chromatids are still connected to each other, as we know. This is during meiosis 1, and the homologous chromosomes have paired with each other. When homologous chromosomes are in this position, some of the chromatids are very close to each other. Then what happens? Part of one chromatid from each chromosome breaks off and reattaches to the other chromosome. This is what happened. Now we have the two homologous chromosomes that pair up with each other during prophase 1 in meiosis 1. Then, in this position, some chromatids are very close to each other and the segments cross. Some of these segments break off and reattach to the other homologous chromosome. If you see, these are the two homologous chromosomes, one purple and one green. Then, at the end of crossing over, part of the one green chromatid, part of it is connected to the part of the other chromatid from the purple chromosome, as you see. Crossing over happens anytime a germ cell divides. In fact, crossing over can occur many times within the same pair of homologous chromosomes. Okay. Because of crossing over results in new combinations of genes, it's also called recombination. The term recombination generally refers to any mixing of parental alleles, including recombination events other than crossing over. Now that you know about crossing over, let's look again at some of Mendel's results and conclusions. Do you remember the results of Mendel's experiment? Okay. As you know from this research, genes located on separate chromosomes are sorted independently, the gene locus. This independence is caused by the random assortment of chromosomes during meiosis. But you also know that a single chromosome can have hundreds of genes. Yes, we have hundreds of genes on each chromosome. This is possible. What happens when two genes are both on the same chromosome? Will they display independent assortment as well, or they will travel together as a unit? Let's see. The answer to these questions is it depends. We cannot say it clearly. Recall that each gene has its own locus, the location or place on a chromosome. Some genes on the same chromosome are close together, others are far apart. Okay, the farther apart two genes are located, they are more likely able to be separated when crossing over happens because they are far apart. Thus, genes located close together tend to be inherited together, which is called the genetic linkage. Linked genes will be inherited in the same predicted ratios as would a single gene. Linked genes means they are close together, so they will be inherited together. In contrast, genes that are far apart are more likely to assort independently because they are far apart. For example, the alleles for flower and seed color are located in the same chromosome in pea plants. This is the flower color and seed color, but they are not near each other. So, because they are so far apart, Mendel observed independent assortment for these traits. As we can see from this picture, this is one chromatid that have 4 chromosomes or 4 alleles, 4 genes. So, gene A, gene B, gene C, gene T. A and B are referred as linked. Why? Because they would likely to be inherited together, because they are closely located to each other. They are close to each other. C and D also referred to as a linked genes because they would likely to be inherited together. But A and B are not linked to C and D. A and B here and C and D here, so they are far apart. Because they are so far apart, crossing over is likely to occur in the space between the genes B and C. So the crossing over might happen here or here, thereby separating A and B from C and D. Genetic linkage let the scientists calculate the physical distance, the actual distance between two genes. By exploring the relationship between them, many genes, scientists have been able to build a linkage map or a genetic map for many species. And this is how we end our section, and thank you very much.